Here's the gel that we'll be loading our hemoglobin protein samples into. We'll be using a micropipette to add the samples to the well. The first sample we're adding is normal blood sample. We'll be putting that in lane 1. In lane 2, we'll be adding a sickle cell blood sample. Notice the crescent-shaped cells. In lanes 3 and 4, respectively, we'll be adding the mother and the patient's blood samples. Pause for a moment here to label which sample is in each well in your student packet on page 40. Be sure to also note what charge is closest to the wells, what charge is closest to the bottom of the gel, and draw an arrow showing what direction the hemoglobin protein will travel in the agarose gel. Then make a prediction about where each protein sample will end up in the gel. Check your responses against the answer key shown here. As you can see, there's a negative charge closest to the wells, a positive charge at the bottom of the agarose gel, and we're predicting that the protein samples will move away from the negative charge towards the positive charge. This is because, as we determined in a previous lesson, the normal and sickle cell hemoglobin proteins carry a negative charge. Normal hemoglobin has a charge of -2, and the sickle cell sample we calculated have a charge of -1. Because the mother sample should be normal, we predict that it's going to run the same distance as the normal sample in the gel, which we see in lane 3. The patient sample is unclear. We're not sure where it's going to end up in the gel because we don't know if the patient has sickle cell disease or some other disease that's causing their symptoms. It's important to note that the hemoglobin proteins will move at different speeds from each other based on their differing charges. The more negatively charged proteins will move the fastest through the gel because they're the most attracted to the positive charge at the bottom of the gel. Before we run the gel, let's make some predictions. Use the differential diagnosis cards posted under announcements on Canvas to complete the chart below. This chart will help you identify what protein is being affected by each of the diseases that we're still considering. Our patient at this point could have sickle cell disease, thalassemia, hemoglobin C Harlem, spherocytosis, or iron deficiency anemia. As you're completing the chart, pay special attention to whether the hemoglobin protein is affected by that disease. If it's not affected, we'd expect the hemoglobin protein to run the same distance as the normal hemoglobin sample through the gel. If it is affected by the disease, we will expect it to appear somewhere else. Check your responses against the answer key shown here. All the information you needed to complete the table could be found in the cause section for each differential diagnosis card. Sickle cell disease has already been completed for you. As you can see, the hemoglobin protein is affected by this disease. It's caused by mutation to the 6th amino acid that turns a glutamic acid residue into a valine. The hemoglobin protein for individuals with sickle cell disease will not be normal. Thalassemia is also caused by mutation to the hemoglobin gene. To date, over 200 different mutations are known to cause thalassemia. Again, the hemoglobin protein will not be normal for an individual with thalassemia. Hemoglobin C Harlem, as the name implies, does infect the hemoglobin protein as well. This is caused by 2 mutations to the hemoglobin gene, one at the 6th residue, which turns the glutamic acid into a valine, just like in sickle cell. There's also a second mutation at the 73rd amino acid, turning an aspartic acid into asparagine. The hemoglobin protein for individuals with hemoglobin C Harlem will not run normally through a gel. Spherocytosis is caused by mutations to the spectrin, ankyrin, band 3, or protein 4.2 gene. Those are all involved in making the cytoskeleton of red blood cells. To date, many different mutations in these different genes are known to cause this disease. Notably, hemoglobin is not one of the proteins affected during spherocytosis. Therefore, hemoglobin should run normally through the gel if the individual has spherocytosis. Iron deficiency anemia is caused by a lack of iron in the individual's blood, as implied by the name. This does not cause the hemoglobin protein to be affected, nor is it caused by a mutation at all. Therefore, if an individual has iron deficiency anemia, their hemoglobin protein should run normally through the gel. Use the information you collected in the chart on page 42 of your student packet to make predictions about what the gel would look like for the 5 diseases we're still considering. Assume the normal hemoglobin protein has traveled the distance indicated in the diagram. Compare your predictions to the image shown here. Notably, individuals with iron deficiency anemia and spherocytosis should have their protein band at the same location as the normal hemoglobin. We predict that the hemoglobin protein of an individual with sickle cell disease will not travel as far through the gel because it's not as negatively charged as the normal protein. Similarly, an individual with hemoglobin C Harlem's protein will travel even less through the gel because it has very little charge to it. It's unclear where an individual with thalassemia's hemoglobin protein would end up in the gel because there's over 200 different mutations known currently. That being said, this diagram showing where the most common form of thalassemia would travel in the band in comparison to normal and sickle cell hemoglobin. Now let's run our gel. When the gel is finished running, draw what you see in the diagram on page 44 of your student packet. The picture on the left shows an actual gel that was run for this experiment as opposed to an animation. Compare this picture to the predictions we made of the 5 diseases that our patient potentially has. Can we definitively conclude that our patient has sickle cell disease and not some other hematological disorder like thalassemia, iron deficiency anemia, hemoglobin C Harlem, or spherocytosis? Why or why not?